Yes. Good morning, everyone. Good morning. Today we have the most expected questions from the chapter Biotechnology. So, all your eyes on the screen. You can have the code for the day on the screen. Yes. Take the quiz seriously because all the questions are very important. Right from level 1, the question levels up to the most important
Take the
question
quiz
itself.
seriously because all the questions are very important.
Yes. The code for today's quiz is 74321. Go to slido.com and use the code 74321. We'll wait for 3 to 4 minutes for everyone to join. Activating the quiz. Let everyone join the quiz. We'll start the session within 2 minutes. Most expected questions from the chapter Biotechnology Rank Booster 2.0, 18 participants and starting the quiz. A dividing and undifferentiated mass of cells is called option A callus, B embryo, C explant and zygote. A dividing and undifferentiated mass of cells is called callus, embryo, explant and zygote. Yes. 88% of the students with the correct answer confirm the answer. Let's have the majority answer on the screen. Breakthrough in genetic engineering has been possible due to the discovery of oncogenes, transposons, restriction endonuclease, and exonuclease. Yes, 5 seconds remaining for the question. Let's have the majority answer on the screen. 85% of the students with the correct answer for the question. Third question on the screen. Agarose extracted from seaweeds find use in option A gel electrophoresis, B spectrophotometry, C tissue culture, and option D PCR. Agarose extracted from seaweeds find yes 100% with the answer gel electrophoresis. Fourth question on the screen. Continuous addition of sugars in fed batch fermentation is done due to option A. Degrade sewage, B. Produce methane, C. Obtain antibiotics, D. Purify enzyme. Yes. The correct answer for the question is option D. Purify enzyme. Continuous addition of sugars in fed batch fermentation is done to purify the enzyme. Fifth question on the screen. Yesterday, many of the students just complained about the time lagging. That's why going a bit faster. Hope you all know the meaning and explanation for all the questions. Which purine is found in RNA? Option A, guanine. C, thymine. D, uracil. Purine found in RNA. Guanine, cytosine, thymine, uracil. The correct answer is option A, guanine. Guanine is the purine found in RNA. Sixth question on the screen. DNA fingerprinting was discovered by James Watson, Alec Jeffries, Frederick Fanker, Hargo bin Khorana. DNA fingerprinting was discovered by James Watson, Alec Jeffries, Frederick Fanker, and Hargo bin Khorana. Yes, Alfred Jeffries is the correct answer for the question. DNA fingerprinting was discovered by Alec Jeffries. Seventh question on the screen. DNA is found in option A, chromosome, B, plastid, C, mitochondria, D, all of the above. DNA is found in chromosome, plastid, mitochondria, D, all of the above. Yes. It is found in plastids, mitochondria, and chromosome because plastids and mitochondria are also contain DNA. Semi-autonomous cell organelle. The correct answer is option D. Eighth question on the screen. Dolly, the first cloned mammal, was produced by option A, parthenogenesis, B, artificial fertilization, C, replacement of zygote nucleus by somatic nucleus, D, in vitro fertilization. Dolly, the first cloned animal, was produced by parthenogenesis, artificial fertilization, replacement of zygote nucleus by somatic nucleus, in vitro fertilization. Yes, 36% with the correct answer. Replacement of zygote nuclei by the somatic nucleus. Ninth question on the screen. Embryo culture is used for option A, establishing suspension culture, B, recovery of interspecific hybrid, C, somatic hybridization, D, haploid production. Embryo culture is used for establishing suspension culture, recovery of interspecific hybrid, somatic hybridization, and haploid production. The correct answer is recovery of interspecific hybrids. Embryo culture is used for, what is the use of embryo culture? It is used for the discovery of interspecific hybrids. Yes. Leaderboard after the question. Yes. Let's check who all are the winners. Yes, we have Vaishnavi 8 on 9, then Guru 7 on 9, Mabashara Iman 7 on 9, Rhythm 7 on 9, and Angelin 7 on 9. 10th question on the screen. First artificial gene was synthesized by Nirenberg, Mentel, Morgan, Khorana. First artificial gene was synthesized by which one of the following? Nuremberg, Mental, Morgan, and Corona. Yes, let's see the majority answer on the screen. Good. The answer is Corona. The first artificial gene was synthesized by Corona. 11th question on the screen. Genetic engineering is option A plastic surgery, B addition or removal of genes, C study of extranuclear genes, D all of the above. Genetic engineering is plastic surgery, addition or removal of genes, study of extranuclear gene, all of the above. Yes, 57% of the students with the correct answer for the question. 12th question on the screen. Yes, another important question. Genomics is the study of genes in general, human genes, genomes in general, and human genome. Genomics is the study of genes in general, human genes, genomes in general, and human genome. Yes, let's have the majority answer. The correct answer is genomes in general. Genomics, read the question. Genomics is the study of. It is not genes in general. It is not the study about human gene. It is not about human genome even. It is about the genomes, all the type of genomes in a general way. You call it as what? Genomics. Perfect. 13th question on the screen. Yes, important question. Important. Uh-oh. Yes, given below is a sample of a portion of a DNA strand giving the base sequence on the opposite strand. What is so special in it? 5 to 3 GAATTC, 3 to 5 CTTAAG. Yes, the answer for the question. Very good. They are the palindromic sequence of base pairs, just exactly like Malayalam. It's a palindromic word like that. GAATTC and CTTAAG are palindromic sequences of DNA. 14th question on the screen. Maximum number of existing transgenic animals is. Maximum number of existing transgenic animal is. Yes. We have pig, fish, mice, and cow as the option. Very good. They are mice. Okay, in the biotechnology chapter, you haven't studied about fishes being used as a transgenic animal. There are transgenic organisms, transgenic fishes are there, but most of the organisms used for biotechnological processes, mice. So, mice is the correct answer for the question. 15th question on the screen. Yes. One of the following process involves southern blotting techniques. rDNA technology, DNA fingerprinting, production of transgenic plants, formation of suspension culture. One of the following processes involves southern blotting technique. The correct answer is DNA fingerprinting. DNA fingerprinting involves the use of southern blotting techniques. 16th question on the screen. Yes, plant tissue culture technique is redefined method of what? Which type of method is a plant tissue culture technique? Is it hybridization, artificial propagation, or vegetative propagation, asexual reproduction, or it is selection? Oh my god. The correct answer is option B, vegetative propagation. So plant tissue culture technique is a well-defined method of vegetative propagation. Perfect. 17th question on the screen. I hope everyone can cope up with the questions and the options, and all explanations are clear for everybody. Polyethylene glycol is fusogenic chemical, electrofusion stimulant, Calus stimulant, differentiation stimulant. Because for after yesterday's quiz, those who were on the YouTube, they might understand that there was so many issues with the internet connection, because every time it appears on the screen, my internet connection is not stable. But I got nearly 10 or 20 messages to start the quiz. It is too late, it is too slow, like that. That's why going a bit faster. The correct answer is fusogenic chemical. So polyethylene glycol is a fusogenic chemical. 18th question, after which you can have the second leaderboard on the screen. Restriction endonucleases are used in genetic engineering and they cut DNA at various sites, join DNA segments, cut RNA at specific sites, cut DNA at specific site. Restriction endonucleases, molecular scissors. Very good. 85% of the students with the correct answer. Guys, what is this? GAATTC, where thymine is present in RNA molecule or an RNA molecule. Have you seen thymine? No, the reverse. The palindrome sequence is CTTAAG. This would be 5 dash 3 dash. Then this would be 3 dash 5 dash. It can never be RNA. This are blunt answers. Okay. Cut DNA at various sites. Then you can use any other enzymes also. What is the function of restriction endonucleases? They cut the DNA at the specific site. You have the diagram in the NCERT textbook also. Sorry, after this question, I'll show the leaderboard. 19th question on the screen. Restriction endonucleases are used in genetic engineering because they can degrade harmful proteins, they can join DNA fragments, they can cut DNA at specific base sequences, they can cut DNA at variable site. Yes, 100% of the question students with the same answer. Restriction endonucleases are used in genetic engineering because they can cut DNA at a specific base sequence. Leaderboard for the question. Yes, we have, oh my god, changes in the leaderboard. We have Rhythm 16 or 19. Then we have Vaishnavi 15 or 19. Ravindra 14 or 19. Ragni 13 or 19. Payal Kaki 13 on 19. Good, good, good. 20th question on the screen. We have 45 questions to complete. Somatic hybridization is achieved through grafting, protoplast fusion, conjugation, recombinant DNA technology. 5 seconds remaining for the answer to the question. Somatic hybridization. Yes, let's see. 79% of the students with the correct answer, it is protoplast fusion. 21st question on the screen. Yes, simple question. Structure involved in genetic engineering is plasmid, codon, plastid, scissors. Plasmid, codon, plastid, scissors. Yes, tell me. 80% of the students with the correct answer, it is plasmid. Guys, you have studied about PB322, PBR322. Studied about like this. These are all our examples of plasmid structures that are involved in genetic engineering. It is never codon. It is never cis. It's never plastid. Okay, 22nd question on the screen. The enzymes required to obtain wall-free naked protoplasm are the enzymes that are used to obtain wall-free naked protoplast are yes, cellulase and proteinase, cellulase and pectinase, cellulase and amylase, amylase and pectinase. Very good. 94% of the students with the correct answer, the enzymes are cellulase and pectinase. 23rd question on the screen. The first hormone artificially produced by bacteria is insulin, thyroxine, adrenaline, testosterone. Eli Lilly company, they have two sequences, A polypeptide chain and B polypeptide chain. Yes, very good. 95% of those with the correct answer, insulin. Let's have the 24th question on the screen. Yes, very important again. The first transgenic crop was the first transgenic crop was? Transgenic crop means those genes who have been altered by manipulation. For example, you have tea, tobacco, flax, and cotton. Select the correct answer among the following. The correct answer is option B, tobacco. Tobacco. Agrobacterium tumefaciens. Meliodignin incognitiae. Perfect. 25th question on the screen. Yes. The most common substrate used in distilleries for the production of ethanol is. Have you all read the question? The most common substrate used in the distilleries for the production of ethanol. You have molasses, corn meals, soya bean, and ground gram. Let's check the correct answer for the question. The correct answer is perfect. It is molasses. 26th question on the screen. The plant Brassica napus have been modified to produce option A vitamin, B hirudin, C lysine rich seeds, and D insulin. Brassica napus. Vitamin A, hirudin, lysine rich seed, insulin. Yes, 47% of the persons with the correct answer. The correct answer is never vitamin A. The answer is hirudin. 27th question, after which you have the next leaderboard. The process of RNA interference have been used in the development of plants resistant to. I told you silencing of mRNA, due to the formation of dsRNA. Insect, nematode, fungi, virus. Perfect. Very good. Perfect answer. It is the nematode. Nematode is the correct answer. Meliodignin cognitiae. I already repeated. So leaderboard after the question. Let's see who all are the winners. Yes, again changes in the leaderboard. Very good. We have Vaishnavi 22 on 27. Then Payal Kake 21 on 27. Rhythm 21 on 27. Shri 20 on 27. Guru 19 on 27. Guru, please improve yourself and come above 20. Guys, the questions are very, very simple and humble questions. And the important questions of the chapter. And again, even the persons who were the toppers in the previous day are also scoring 22-23 range. I wanted the topper to score about 25 after the third leaderboard. Because you can lose two questions because sometimes they may be out of NCRT or they will not get strike to your mind immediately within this 15 seconds of answering. But 22 is a poor score, 21, 20, 20, 19. Then I can imagine the condition of the rest of the 17 people who are playing the quiz. Below 15 only they might be having, I don't know. Yes, 28th question on the screen. The specific triplet of nitrogen bases on the transfer RNA is called codon, anticodon, genetic code, none of the above. If I got the time, I would have explained each answers and would be going. But yesterday, many students were complaining even in the Slido platform itself that time consuming, time is important. The answer is anticodon. That's why I just repeat the important points and I'm just going. Not going into the depth of each and every question. But when you observe the last test, that is the last Slido quiz on the chapter, what was the last topic that we discussed yesterday? Biological classification. I've explained each and every point along with the diagram. And most of them told that it was time consuming. Okay, the specific triplet of nitrogen bases of RNA is called anticodon. 29, the question on the screen. Yes. The term gene was coined by M.C. Clintock, Morgan, Johansson, D. Dewey. The term gene was coined by. I'm asking about the term gene. Gene is the hereditary unit. In order to avoid the time, I'm just explaining now. The hereditary unit. Let's check the correct answer for the question. The correct answer for the question is Johansson. Please bookmark this. This was 2004 AIMS previous year question. We have NCRT Challenger coming up with the name of the scientist. Okay, you will be having one more slider quiz sharp at 11 a.m. today. Sharp at 11 a.m. you will be having the NEET Abhyas paper test number 1 discussion on the slider platform. Yes. 30th question on the screen. The transgenic animals are those which have, I already told you will be having at the 11 o'clock, 11 a.m. today, you will be having the NTA paper, NTA mock test paper number 1 discussion from BioPoint, Stream the World of Bio. So stay tuned with the Telegram group for the link and the code. Transgenic animals are those. Correct answer by 42% of the students. Transgenic animals are those which have foreign DNA in all their cells. 31st question on the screen. Next leaderboard is after the 36th question. The two strands of DNA are held together by. The two strands of DNA are held together by nitrogen, oxygen, hydrogen, carbon. Suppose this is a one strand of DNA. This is another strand of DNA. Yes, perfect. 100% of the students with the correct answer. If this is adenine, this is thymine, you will have the what type of bond over here? You will have the hydrogen bond. Perfect. You have the hydrogen bond over there. That is the correct answer for the question. 32nd question on the screen. There is a restriction into nuclease called ECOR1. What is co part in it stands for? Is it for coli? Is it for colon? Is it for xylome and coenzyme? You have the term as to be ECOR1. Perfect. What is the students with the answer coenzyme? It is not coenzyme, it is coli. E stands for the genus name. It is Escherichia. CO stands for the species name. It is coli. R stands for the strain. And 1 stands for the number of discovery or observation. Perfect, everybody. So the correct answer for the question is option A, coli. 33rd question on the screen. Transgenic plants are developed by introducing foreign genes, introducing gene mutation, deleting certain chromosome parts, stopping spindle formation. Transgenic plants are developed by. The correct answer is given by 100% of the students. Transgenic plants are developed by introducing foreign genes. I hope there must be some changes in the leaderboard because most of the students are giving the correct answer for the questions. 34th question on the screen. Cry genes or BT genes are obtained from. Very easy question. Cotton pest, tobacco plant, Bacillus thuringiensis, and E. coli. I'm asking about the crystal gene or the cry genes and the BT genes. BT. What does the term BT stands for? Guys, is it cotton pests? Never. Is it tobacco plant? No. Is it E. coli? No. You have the BT. What is it? Bacillus thuringiensis. Exactly, option C is the answer for the question. 35th question on the screen. Which of the following enzyme joins the Okazaki fragments? The question is about the Okazaki fragments. Is it DNA polymerase, DNA ligase, helicase, and restriction into nuclease? Hope everyone voted for the question. Yes, the correct answer is DNA ligase. Restriction endonucleases. Please remember, restriction endonucleases act as the molecular scissors. Molecular scissors. It cuts the DNA at specific fragment. DNA ligase act as a molecular glue. They join the different DNA fragment. They help in the joining of different DNA fragments. Yes. 36 question, after which you have the fourth leaderboard. I hope 34 plus marks for the topper. Which of the following features should be present in a vector? It should replicate autonomously. It should have its own origin of replication. It should possess restriction enzyme cleavage sites. All of this. Why no comments in the Slido quiz? I don't know today. I think all are busy with their work. Yes, all of this. So, what are the characteristic or the major characteristic for a vector? It should replicate autonomously. It should have its own origin of replication. It should possess restriction enzyme cleavage site. That is the answer should be all the above. 95% of the students are reading and writing or selecting the answer correctly. After this question, leaderboard. Which of the following is a genetic vector? Plasmid, phage, cosmid, all of this. Genetic vector. Plasmid, phage, cosmid, all of this. Very simple question. Simple question. Yes. The answer is all the above. Is a genetic vector. You know that plasmid is a genetic vector. You know that phage is a genetic vector. You know that cosmid is a genetic vector. So this one, and the third one also comes under what? Genetic vector. Am I right? It also comes under genetic vector. So the correct answer is all of these. Clear? Perfect. Leaderboard for the question. Yes. Very nice changes. Very good. Everybody's studies are going down day by day. We have Shri 30 on 37, Payal Kaki 30 on 37, then Vaishnavi 30 on 37, Rhythm 29 on 37, Priyadarshini 27 on 37. Very good way of study, children. Nearly 30 days remaining for NEET 2020 and the most important chapter, biotechnology, application, principles and processes. Let's have the 38th question on the screen. Read the question correctly and answer the option, okay? Which one contains 4 pyrimidine bases? You know that there are two type of nitrogen bases, purines and pyrimidines, contain at least 4 pyrimidine bases. They're asking about 4 pyrimidine bases. The correct answer is option A. 39, the question on the screen. Again, simple question. Bacillus thuringiensis is used to control nematodes, fungal pathogen, bacterial pathogen, insect, pest. Yes. Guys, it is insect pest. Why is the answer nematodes? Nematodes. Meliordignin incognitiae in case of tobacco plant. Using Agrobacterium tumefaciens. Agrobacterium. Nematodes can never be the answer. It is never fungal pathogen. It is never bacterial pathogen. The answer is insect pest. Cotton plant you are taking. Bollworm. Corn borer. I think you remember all these things when I at least tell the names. 40th question on the screen. We have 5 more questions. The genetically modified GM brinjal, genetically modified as GM. Brinjal in India has been developed for enhancing shelf life, insect resistant, drought resistant, enhancing mineral content. Yes, the correct answer is insect resistance. Guys, genetically modified brinjal, enhancing shelf life, no. Drought resistance, no. Enhancing mineral content, no, it is for mostly it has been developed. All these functions are there in your NCRT. I remember, I know that. The most appropriate answer when this type of a question approaches, you have to select the most appropriate answer. You have to select the most appropriate answer for the question. Yes, 41st question on the screen. The protein products of the following Bt toxin genes, CRY1AC and CRY2AB, are responsible for controlling. Is it bollworm, roundworm, moth, and fruit fly? Those who have read the NCRT might. Yes. Guys, you haven't read about roundworm. You have learned about only bollworm, cotton borer. 42nd question on the screen. Question from molecular basis of inheritance comes in biotechnology processes also. VNTRs, variable number of tandem repeats in the DNA molecule, are highly useful in monoclonal antibody production, DNA fingerprinting, recombinant DNA technology, stem culture technique. The correct answer is option B, DNA fingerprinting technology. In the chapter molecular basis of inheritance, you studied about the VNTRs, variable number of tandem repeats. In case of DNA fingerprinting, there you haven't studied about the monoclonal antibody production. It's not about recombinant DNA technology or stem tissue culture. Yes. 43rd question. You have two more questions remaining. Restriction endonucleases are widely used in recombinant DNA technology. They are obtained from bacteriophages, bacterial cells, plasmids, all prokaryotic cell. Yes, 50% of the students with okay. Restriction endonucleases, guys, restriction endonucleases obtained from E. coli. E. coli, Escherichia coli, it's a bacterial cell. It is never a bacteriophage or a plasmid or a prokaryotic cell. Yes, two more questions. We have the final result. Only 3 comments yesterday. There were 27 or 31. Yes, what happens? PCR proceeds in 3 distinct steps governed by temperature. They are in the order: annealing, synthesis, denaturation, synthesis, annealing, denaturation, denaturation, annealing, synthesis, denaturation, synthesis, and annealing. Yes. 84% of the students with the correct answer for the question. It is denaturation, annealing, and synthesis. You have the last and the final question of the most expected questions, Rank Booster 2.0 from biotechnology on the screen. Yes, the final question, bomber question to determine the leaderboard on the basis of the timer of the question. Sterilization of tissue culture medium is done by mixing the medium with antifungal agents, filtering the medium through fine sieve, autoclaving of medium at 120 degrees Celsius for 15 minutes, keeping the medium at minus 20 degrees Celsius. Guys, read the question. Sterilization. How can a minus degree temperature comes over there? Always sterilization means boiling in water. Only the temperature, high boiling temperature given is autoclave method. You might have heard about the autoclaving system used in the hospitals to purify the instruments in the operation theater. Right? So the final leaderboard on the screen, you have the winner to be. Yes, let's have the winner. Yes, very good. Congratulations. You have Payal Kakade, 38 on 45, not even crossed 40 marks the topper. Okay, I'm not satisfied with today's result. Moreover, the first position for Payal Kakade, 38 on 45, then Shri for 35 on 45, Vaishnavi 35 on 45, Ragni 34 on 45, Rhythm 34 on 45. The hardest question answered by only 6% of the students. The first artificial gene was synthesized by. Yes. I'm deactivating the quiz. Bye bye. Take care. See you sharp at 11 a.m. with the NTA mock test, NTA Abhyas mock test number 1 paper discussion in Slido. Stay tuned with our Telegram group, BioPoint, Neat Extras, PCB doubts for the code and the link for the session. Please don't forget to like, share, and subscribe the wonderful session. Deactivating the quiz. Thank you, everybody. Oh, today also I got start fast and anonymous. Actually, I haven't seen him on the leaderboard at least. Was keenly interested in playing the quiz. Yes. Anyone else? No. Thank you, everyone. See you. Take care. Bye bye.